So good afternoon, everyone. I am Arkaprava Banerjee from DTC Lab, and today my topic of presentation will be the development of a global Q-RASAR model for the efficient quantitative predictions of skin sensitization potential of diverse organic chemicals. Now, in the environment, there consist of various kinds of compounds that can cause a wide array of toxic effects, and one such toxic effect can be the institute of skin sensitization potential. Now, in the past, various QSAR models have been developed, but most of them have been classification-based models. Some of them were regression-based models, but they were only concentrated on a specific type of reaction mechanism that we have shown on the right-hand side. Now, this study aims at developing a global Q-RASAR model built using diverse structures, irrespective of the mechanism of action on the skin. So the concept of Q-RASAR comes from merging the concepts of QSAR and read across. So in QSAR, we compute various structure and physicochemical descriptors that can be used for QSAR analysis. Additionally, we compute similarity-based descriptors that can be used for read across. Now we combine the structure and physicochemical features with the similarity descriptors to obtain a complete descriptor pool from where we can perform feature selection and develop Q-RASAR model in a QSAR framework. Now, what differentiates Q-RASAR from QSAR is that the QSAR descriptors are computed based on the whole data set, whereas the Q-RASAR descriptors are computed based on the training and test set separately. Second thing is that the QSAR descriptor for a particular query compound represents the chemical information of that particular query compound, whereas the RASAR descriptor for a particular query compound represents the information of its close source neighbors for that query compound. Now, this is the basic workflow where we have collected the data from different literatures. We have curated the data, descriptor calculation, data pretreatment, data set division, feature selection, and we have developed the PLS QSAR model using the TTC QSAR tool. Additionally, we have optimized the read across based hyperparameters. We have computed the RASAR descriptors. We have fused the initially selected structure and physicochemical features with the RASAR descriptors, and we have detected the prediction confidence outliers from the test set, from the DTC plot. We have removed them from the test set, and we have developed PLS QRASAR model. Additionally, we have employed various machine learning algorithms to compare the prediction ability. In addition, we have developed a Java-based sample tool which can take the input of a query compound and it can calculate the skin sensitizing potential along with its applicability domain status in terms of leverages. Now, this is the novel DTC plot to determine the prediction confidence outliers of the PLS QRASAR model. So the y-axis represents the absolute difference in the response value of the first two closed source compounds, and the bubble diameter represents the absolute difference in the similarity values of those closed source compounds, and the x-axis represents the highest similarity value to the closest source compound for each query compound. Now, in contrary to the property of the similarity property principle, two compounds having a large difference in the activity should ideally have a large difference in the similarity values as well. So a bubble having a larger bubble diameter should ideally be located higher in the y-axis, but bubbles having smaller diameters located higher in the y-axis represent that they do not follow the similarity property principle, and thus these compounds can be identified as outliers. So this is the case where a particular query compound, where the similarity values are not quite as high and the response difference is also low. But in case of DTC plot outliers, you can see that the similarity values are low, but the response difference is quite high. So this compound can be removed from the test set. So this is the PLS QRASAR plot where the features like presence of conjugated double bond, presence of aromatic carbonyl group can actually help in skin sensitizing potential. And the values of R-squared chain and the cross-validity Q-squared represents the model is highly robust, and the value of MAE test represents the model is highly predictive as well. So these are the 3D surface plots for two of the most important descriptors, SPTO and NCSP2, where it is a 3D plot representing the predicted residual with the experimental PEC3 value and the descriptor value. So we have additionally divided a data set into different clusters by the k-means clustering technique. And for each cluster, we have used the model to make predictions and derive MAE values. Now, in most cases, you can find that the MAE values are medium to low, suggesting the global performance of our model in each cluster. Additionally, the values of the descriptors are represented in the form of a heat map from where we can understand the positive and negative correlation of the descriptor. So this is the comparison of predictivity among different ML models, where the lowest MAE test has been represented by the multi-layer perceptron model, which is just lower than the corresponding PLS model. But in terms of simplicity, interpretability, and reproducibility, we infer that the PLS model is the best over here. So this is the SHAP analysis plot of the different descriptors for the MLP model for the training and test set. And on comparing our PLS QSR model with the PLS Q-RASAR model, you can see that there has been a significant increase in the Q-score F1, F2, and ME test values, suggesting enhanced predictivity. And the values of R-square train and Q-square are similar, suggesting similar kind of robustness. Whereas in previous works, you can see that they have used a lot lower number of compounds. Whereas in works like Dearden and his team, they have worked on a large number of compounds, but they haven't reported any kind of external validation metrics. And moreover, the values of R-square train and Q-square LO, they do not meet the threshold of 0.5. This is the expert system that we have developed, and it is available from the DTC Lab tool supplementary site. And lastly, I'd like to conclude that a simple, reproducible, transferable, robust global PLS Q-RASAR model has been developed. The MLP Q-RASAR model shows the best predictivity, but in terms of interpretability, reproducibility, and transferability, we infer that the PLS Q-RASAR model is the best, and a Java-based expert system has been developed for the quick and easy prediction of skin sensitizing potential. Thank you.
Please, next. There is some questions from Vladimir Paroikov. How your model could be applied to so-called activity cliffs?
Activity cliffs.
Yes.
Yes, we have actually done this with a classification-based data in our previous publication, where we have proposed two novel similarity-based metrics, obviously the RASAR descriptors, and those descriptors can actually identify the presence of activity cliffs in the training and testing based on the descriptor value itself. Means we do not have to develop a model.
There is another question from Dmitry Shulga. Why the stats for the test sets is significantly higher according to your plot? Could you explain this?
Yes, in our previous work, if you follow the RASAR modeling, you can see that there has been a trend in most of the cases that the external prediction quality is higher. This is because in QSR, we compute the descriptors based on the whole data set with the number which represent the property of the closest of the particular query compound. But in case of RASAR, we actually compute the descriptor based on the closed source neighbors from the training set. Therefore, we are trying to extract the information from the training set and trying to embed this in the test set. That's why probably it shows higher prediction. Moreover, in case of training set RASAR descriptor calculation, we have applied the leap same out algorithm. That means while calculating the training set descriptor, while we are taking the training set as the standard one and the query set as the training set itself. So therefore, for the each identical compound is omitted. That's to reduce the overfitting of the model. And that's why in most cases, we find that there is a slightly reduced internal validation matrix for RASAR models over QSR models, but here it is comparable, which is good.
Roman Yefremov asked, how consistent was DB on the sense data taken from the different sources?
Sorry, I couldn't get the question.
How consistent was DB on the sensitivity database
yes,
from
we have
different sources?
Yes, we have obtained this data from previous literature from the works of Nash and Jeffrey, and this is the skin sensitizing data for the local lymph node assay of murine species.
And another question from Professor Sulimov: was an experimental testing made?
No, there hasn't been any kind of experimental testing. Actually, it is the local lymph node assay data for which was actually reported in some other works. So we have taken this data and we have developed a model. The basic idea behind developing this model is that in the literature we find that there are various skin sensitizing models, but each of them are concentrated on a particular type of reaction mechanism. Say, for example, some models are just concentrated on the Michael addition reaction. Some models are concentrated on the sheet-based formation. But we have tried to encode those diverse structural features into a single model where one can easily predict the skin sensitizing potential without knowing the reaction mechanism of that particular query compound.
Since you didn't make experimental testing, how you prove your model?
Since we have, yes, it can be proved. If you just get the sort file, that means in case of RASAR descriptors, there's another file generated which shows the closed source compound. Now, the closed source compound comes from the training set, which have actually experimentally tested values. So from the closed source compound, say, for example, we have 10 different closed source compounds. So if we have 5 compounds that show active action using the Michael addition reaction and 3 compounds showing sheet-based formation and 2 compounds showing which are acylating agents. So we can identify based on the number of closed source compounds that are showing a greater number that are showing a particular reaction mechanism. Moreover, we can also identify the highest similarity closed source compound.
I don't ask this anyway. We have to stop because we are out of the time. Thank you very much.
Thank you.
The next
Thank you.
speaker.
Athena, sorry.
Yes.
We are ahead the schedule. Look on the clock on the right
I
high
have, yes,
part.
but I have my clock. According to
No,
my,
you have,
okay.
but we
Okay.
are ahead the schedule. So we
Okay.
may be a little bit more slowly, not in
Ah,
this way. Please.
Okay, so please continue with your answer.
Yes, so if you just look at the closest source the similarity value of the closest source compound, so that source compound shows a particular type of reaction mechanism. So we can also infer that the particular query compound would have a similar reaction mechanism as well.
Thank you.